Now we're going to look at CNS neuroinflammation, because we know that chronic inflammation is one of the hallmarks of MS, and it's the reason for axonal degeneration and myelin degeneration. So therapies aimed at decreasing the expression of pro-inflammatory pathways and improving the expression of anti-inflammatory pathways are essential in helping MS. So some of the here we go. So this is important. Cytokines are proteins, are signaling proteins that mediate immune response. You have pro-inflammatory cytokines and you have anti-inflammatory cytokines. So some examples of the pro-inflammatory cytokines are interferon gamma, interleukin beta, interleukin 2, interleukin 12, interleukin 17, and some examples of the anti-inflammatory cytokines, which we want more of, interleukin 4 and interleukin 10. So we're going to look at some animal studies of CNS neuroinflammation. We know that there's a tendency toward exercise decreasing pro-inflammatory cytokines while increasing anti-inflammatory cytokines. This is shown in other research, but we can extrapolate it and hypothesize this happens in MS patients, but they're going to go on and use animal models to further provide some evidence. So during neuroinflammation, I explained before, there's infiltration of peripheral immune cells into the central nervous system as the blood-brain barrier integrity becomes worse. So the blood-brain barrier is a protective mechanism where you have a tight junction in the capillary, and some of the neurons form foot processes over the capillary to prevent external toxins and immune cells to enter. But in MS, we have a breakdown of that blood-brain barrier and the peripheral immune cells enter the central nervous system. But the process of which requires the attachment, the process of the peripheral immune cells entering the central nervous system requires the attachment of immune cells to various adhesion molecules. So this is interesting because animals that were allowed to exercise prior to MS induction displayed an increase in tight junction proteins, and these proteins are called occludin and claudin 4, and a decrease in the adhesion molecules PECAM1. So remember, we said that the peripheral immune cells need to adhere to molecules to enter the central nervous system, and this study showed that animals that were exercising prior to MS induction had a decrease in these adhesion molecules. So overall, we had a reduction in blood-brain barrier permeability. This was a study in 2017. So this reduction of blood-brain barrier permeability also showed a decrease in infiltration of B cells, T cells, and monocytes into the spinal cord of exercising EAE mice. This was shown in, they've listed 5 different studies here that have shown this same thing. So exercise in MS models of mice decreased the blood-brain barrier permeability to these pro-inflammatory molecules. So microglia are the phagocytes. They're the main immune response in the central nervous system, and they respond to injury and inflammation, and they secrete cytokines. So that's what I just said. Okay. So what the article goes on to say here is that microglia are a very vast type of neuron in the central nervous system. There's too many different phenotypes or subtypes to classify, so they're just going to break them down into 2 different categories. You've got the M1, which are the M1 reactive-like microglia, and these release pro-inflammatory cytokines, so we want less of these. And then you've got the M2-like microglia, which are anti-inflammatory. So we want more M2-like and less M1-like to have a greater balance of anti-inflammation in the central nervous system to benefit MS patients. So there's a study here in 2018 where they found that M2-like microglia, the anti-inflammatory, are involved in the attenuation of inflammatory response by removing inhibitory factors, decreasing toxicity, and increasing the release of anti-inflammatory cytokines. And they also found that active MS plaques are often populated by M1-like microglia, and remember, M1 microglia are the pro-inflammatory. So if we can show that exercise reduces these, then we can show that it has a positive effect on MS through reducing inflammation. So a study in 2013 showed the effect of exercise on microglia in exercising mice had a higher number of M2-like regulatory microglia and a high number of insulin-like growth factor, which authors suggest to be a neuroprotective phenotype. So we see here in a study in 2013, we have an increase in anti-inflammatory markers. And they also found that there were significant decreases in the M1-like phenotype microglia observed in the striatum and the corpus callosum of demyelinated treated mice after 3 weeks of voluntary wheel running. So we saw this study is saying that 3 weeks of voluntary wheel running, aerobic exercise, decreased the amount of M1-like microglia in the striatum. So the striatum is one of the parts of the brain that communicates with the basal ganglia, and it's involved in regulating voluntary movement. And again, the corpus callosum is the middle segment of the brain that connects the two hemispheres, so they can communicate with each other. So here we can see that exercise has a positive effect on neuroinflammation. And we've got some human studies. So basically, the human studies of neuroinflammation look at the peripheral effects by looking at blood serum levels and plasma levels. And there's a strong evidence that exercise can reduce the permeability of the blood-brain barrier. Then we go on to neuroprotection. So we've gone through oligodendrocytes, we've gone through remyelination, and we've gone through neuroinflammation, and exercise has a positive effect on all three of these in animal models of MS. So what's neuroprotection?